malaria and dengue control.
Thank you. You've already heard that dengue is one of the forgotten diseases. It's clearly forgotten enough that the typist couldn't spell it on the abstract. But I'm just going to start off with perhaps a couple of contentious sort of statements here. And the probably, I guess, is the contentious bit on this first statement. Probably every 30 seconds a child, and that's primarily in Africa, dies of malaria. Now, I say probably because you've already seen the hippopotamus and you've already seen that 80% of the deaths of malaria occur at home. How on earth do we know how many of those really are malaria? We don't even have good statistics for how many people, how many children really are dying. And I was pleased to see that at least the two numbers you've had so far were 1.08 and 1.09 million, but those estimates in fact range from about 1 to 2 million children per year dying. So if you can't even get the estimates like that right, how on earth are we actually going to actually go on and cure the disease? And then dengue, again, one of the fastest growing viral diseases. This is a disease that's on the move. It's on the move primarily because of humans, and it's on the move because the vector that transmits it lives in very close association with man, and we're very good at sending it around the world in tyres and various other sort of bits and pieces there. So we're causing our own problems there. Two very big differences between the two diseases that we're looking at here, and you've already heard the great excitement, and I would share that, of the Anopheles genome being sequenced. Well, what we've actually sequenced is the Anopheles gambi genome. That is one of several hundred mosquito species that actually transmits malaria. Yes, it happens to be an extremely good one and one of the major African vectors of malaria, but it's not the only African vector of malaria. And in fact, again, you heard part of Carlos's talk that the Anopheles gambi mosquito homes in on humans no matter what else happens to be around to bite. Anopheles funestus, which is the other major vector of malaria in Africa, equally well homes in on malaria and is probably actually a better vector than Anopheles gambi. And you've only got to look at the transmission data that we've had fairly recently from Mozambique and South Africa to realise that. With extremely low numbers, you're getting very high transmission rates relatively as we go through. So I think there's a caveat to be put there in terms of what we've got so far. Dengue, on the other hand, is primarily transmitted by one mosquito. It's moving around the world, as I say, very quickly, but it's Aedes aegypti that transmits. And we've got a secondary transmitter, which is Aedes albopictus. But if you haven't got the aegypti there, then albopictus isn't going to cause you major problems as you go through. So that's just a little bit of setting for where we're going at the moment. What tools do we have? Well, we've got insect vector control, or if you really want, you can splash repellent on you, and that makes you smell nice with diethyltoluamide, or probably citronella if you want a prettier smell as you go through, and drugs for prophylaxis or for treatment. Now, just to show you the progress that we've made, again, you've heard that between 1955 and 1969, we went for malaria eradication. We had a magic bullet. We had DDT. DDT was going to get rid of malaria for us for good. Would anybody like to tell me what South Africa, Swaziland, Mexico, India to an extent are currently using for malaria control? DDT. 50 years on from now, that same spraying of a compound that is now well over 50 years old is the primary cornerstone of those malaria control programs. And those countries are not the poorest countries. If you look at the list that I've just given you, those are the ones that can actually afford to put the control programs in. Many of the poorer countries have got nothing in there. I'll come back to that in a minute. There's a reason for some of that DDT problem still running through. But I think that is, again, something to keep in mind as we go through here. We look at malaria. Who's sort of setting the agenda and what are we going through? Well, it's great to see actually malaria control is back on the agenda. We've got the Millennium Development Goals, the Abuja malaria targets, rollback malaria phase 2. We're actually getting it out there into the populations rather than talking about it. The donors' agendas, because a lot of this is driven by the donors and local expertise and the collaborators that we're working with. But we need to put all that together if we're actually going to make a real difference for malaria control as we go through. Current directions and priorities. Well, I'm being politically correct here, but I'll be politically incorrect in a minute. In terms of saying we can reduce transmission and we can go for integrated vector control. It's wonderful. Doesn't work. Nobody does it. What we really do is residual house spraying, if you really want to know what we do by way of integrated vector control most of the time. Or if we can't afford residual house spraying, we put our insecticides on bed nets, but when we do that, we actually reduce the number of insecticides that we've got that will work, because at the moment, we can only put one class of insecticides onto bed nets, and that's pyrethroids. We can do that, A, because we've got to make sure that the human toxicity is pretty low if you're putting things that people are going to sleep underneath. B, if you're going to actually protect the person who's sleeping under the net, you've got to kill that insect fairly quickly. Otherwise, any of you who've slept under a bed net will know that the mosquito will sort of land on there, and if your arm's on the side of it, it will bite through the net unless that insecticide really is going to knock it down very quickly, or you're going to have something that the insect doesn't like and it's going to repel it. And there are very, very few insecticides that actually sit with those characteristics, and effectively, pyrethroids are it. That is the one sort of set that we've got. Now again, let me come back to monotherapies. How many times have you already heard that monotherapies are bad things? And what are we doing by rollback malaria in the primary sort of set? We're going in with a monotherapy. We're putting pyrethroids onto bed nets and saying, hey, here we go with another magic bullet. It doesn't work in South Africa, and that's why we've got what we've got going on there with DDT, because they've got a lot of resistance coming through with the pyrethroids. Prevent severe disease. We need to identify the vulnerable. We need to diagnose accurately, and we need to treat early. These things are things that we really should have been able to say 40 years ago. We probably could. How well are we doing then if these are obvious things? Because they really should be obvious to most people. I'll go through some of those in a minute. But on from there, we need capacity building if we're going to actually work out how we're going to control malaria as we go through, or dengue for that matter. We need the people on the ground with the vector control sort of capacity. It's got worse over the last 20 odd years that I've been working in this field, not better. There are fewer people now in Africa who actually could identify a mosquito to species level. And in fact, we've got a little bit better on the resistance side, because over the last sort of 5 years, I've personally been involved in helping training 75 people across an awful lot of Africa. But before that, when we started off putting MIM networks into place, there was nobody in most of Africa, I could have counted them on the fingers of one hand, who could actually go in and do a decent resistance test for you and interpret it as we went through. So we haven't done brilliantly on the capacity side, and I think we need to do more. And in terms of understanding the prophylaxis and treatments that we're putting in, combination therapy is clearly coming in with the drugs on the malaria side, and I welcome that, but we need to understand what we're doing, why we're doing, and how we're doing it. And then on to the new knowledge, and here's the sort of crystal ball bit as we go through. Vaccines, drugs, new insecticides, transgenics. Brilliant new technology. A lot of us are very excited about that, and that includes me as we go through. But if we're going to do our jobs, it's not just developing those tools and technologies, it's getting them out there, working with the scientists in the developing countries to actually put these together and actually implement them in a way that's sensible. And I worry an awful lot that we're not going to do that, because there are all sorts of competing pressures on us that make it extremely difficult to do that as we go through. So let me pick up on some of those sort of bits that I've gone through so far. You'd think by now it would be relatively straightforward to diagnose malaria. It is to some extent, but what or how do most of us diagnose malaria who are actually out in the field? You've seen pictures of some of the clinics that go on there. Most of us who are diagnosing malaria out in the field, or most of those whose job it is to diagnose malaria out in the field, will do a clinical diagnosis. That means they will generally ask people if they've had fever, and if they have, they'll have their malaria tablets and they'll be sent off home. There are other ways of doing it. You can clearly diagnose malaria by PCR, you can do it by antibody detection, antigen detection, microscopy, or the clinical diagnosis that I've just given you. Under many field settings within Africa, the first two of those are not practical. Antigen detection is getting used more. Microscopy is the gold standard, but it takes time and it takes people who know what they're looking at. And again, there aren't huge numbers of people out there in many of the clinics in Africa who could actually do a decent diagnosis using microscopy slides, even if they had the microscopes to do it. And I've travelled around a huge number of clinics in Africa that don't have the microscopes to actually do that. Do we have any data that says clinical overdiagnosis is going on? And what's that doing in terms of the drug wastage that's going on out there and drugs not being used properly as we go through? Here's just an example from Swaziland, separated by age ranges. Here are the correct diagnoses out of the numbers that are actually done. These were done by that gold standard microscopy after the general diagnosis had gone through the clinics. And you can see that 73% of the 0 to 4s were being misdiagnosed. It got a little better in the 10 to 14-year-olds and back up at 80% here. And these are in very large numbers. So clearly in Swaziland they weren't doing too well, but maybe that's just the odd exception. Well, here's a little bit of data from Namibia. In the dry season, a whopping 92% of the samples that came in had been misdiagnosed. They were not malaria. They'd been given malaria tablets, told they had malaria. This was in the dry season when you're not getting a huge amount of transmission anyway. In the wet season, they did a bit better. About 50% of those were correctly diagnosed. I could go on. These are not exceptional data. So there's a huge problem already out there with diagnosis. Okay, so we get the diagnosis a bit wrong. But what about this early detection sort of set? And are we getting that right? And are the drugs really getting where they need to go? Here's just a quote from a lady out in Ghana. And you can again read it for yourself. We're back into the inequitable access to malaria care. Basically, people don't go from the villagers, the poorer people, to get treatment until they're into the late stages of malaria. They feel they can't afford to do that. What do? They take herbs and drugs from the local drugstore because they're more easily available, they're cheaper, and they can't finance the illness. So what happens when they do go and get treatment, and what makes them seek treatment? Well, clearly the severity of the illness. They wait until their children get sick. They wait until their children get really sick, and then they walk long distances often to hospitals. And before they do that, they have to go and borrow the money they know they're going to need to pay for that treatment when they get there. And there are long discussions about who's going to pay and how. And another quote from the same village, basically, lady there, has treated her malaria for 4 weeks with paracetamol and herbs. It hasn't got better. She's just got more and more ill. And then she's had to spend 10 weeks' wages going to the hospital and getting treated for anemia because the malaria has got to that stage. So problems around with the treatment there. What about improving the quality then of the laboratory services and getting that diagnosis in? Can we do that? And where are the problems there? Well, we can try and ensure that performance of all laboratories is up to a certain standard. We can try and establish standardized regional laboratories and educate clinicians to make the best use of those laboratories when we put them in. And there's been a very nice study in Ghana that's been doing that ran from 2000 through into 2001, which was set up as a baseline survey initially of all government laboratories, and they weren't in great shape when that was started off. 95% of the staff were then trained in 11 basic tests, and quality control measures were actually put in to do that. And an awful lot of that was looking at accuracy then in looking at hemoglobin, malaria, and the other sort of basic sort of techniques that they were needing to put in. And that's now been rolled out after being very successful to Malawi, where we've had technicians visiting that sort of program. But this is integrating the services and trying to put TB, HIV, and malaria through central laboratory services to at least make the best use of the resources that we've got out there, because I'll show you some data in a moment. These are not hugely well-resourced laboratories as we go through. They're often running with very small budgets as they go through. Here's the sort of thing that was being done in the Malawi District Hospital And a lot of this was malaria, but blood transfusion is coming down here that linked up to malaria. So you can see that a fairly hefty burden is coming into those labs from malaria. And if we split that down by cost, and the total cost of this lab's about $32,000 a year, less than the cost of a postdoc working in my lab, certainly, to put that into to context. Then again, the huge amount of the cost that comes through there is linked up to the blood transfusion screening and malaria sort of set. Can we do anything to improve what's happening with the mortalities of the under 4s or under 5s? Because we've already heard that a problem is actually getting to treatment fairly quickly. These kids are not getting to the hospitals and clinics until they've got real problems with anemia as they come through, because the treatment isn't being sought at that level. Well, a few of the examples you've already been given from Carlos, I'm not going to repeat in great detail, but part of that is clearly tying things up to the vaccine sort of side and giving these kids treatment when they do come in for the vaccines with prophylactic doses of drugs. It's also trying to keep their iron and hemoglobin levels high by giving them those. But if they come in with severe anemia, can you do anything about it? Because again, the reality is if these kids turn up at the clinic and they're in bad shape, you've really got 24 hours to do something about it by the stage most of them come in. Because if you don't up their hemoglobin counts in 24 hours, you've lost them, those kids are dead. Now, we've got a real problem then if we're looking through, because most of these sets, you're not working with decent blood banks. They don't have the kind of donor programs that we have in Europe or the US with great screening programs for HIV. So you need a system that's actually going to allow us to treat these kids quickly with blood that's safe if we're going to do that. And one of the sort of small studies that we've had running of late is to look at the feasibility of actually collecting cord blood for transfusions from the district hospitals from normal births. And those are from births which are then screened for HIV, so we know that those are actually safe as they've come through. The study's actually been done again in Ghana at a teaching hospital there where they have 11,000 normal deliveries each year. And again, just to give you a little bit of statistics on the malaria sort of side as they come through, 90% of the children who come into the hospital there are anemic. 40% of those children have severe anemia, and you're going to get 40% mortality of those if the hemoglobin count drops. And as I say, you only get 24 hours really to have a shot at this. We also know that the role of malaria has in childhood anemia, and that peaks at 12 months of age, and that's exacerbated by infection and poor nutrition. As I say, there are problems getting the blood. The old system of loaning from relatives is a slow process and made much worse since HIV, and we don't have the blood banks to go in for. So can we actually sort of use that cord blood in a simple system of collecting it? Well, here's the system that it's collected. You just take the placental cord and you let the blood drain normally out of the set into the sort of bags that we've got, and that is then just sort of stored from those normal uncomplicated deliveries as we go through. That blood collected in that format with no fancy equipment, no nothing else, from 18% of the normal deliveries given to most of those children who come in will actually improve the chances of survival in 65% of those children that come through. Now that's not fancy technology, but it's just trying to put some sensible technology into the sort of system. I've sort of told you a little bit of what we can do and the problems that are already there, and I haven't even touched on the fancy genomics yet. And I'm sitting here, I guess, as a sort of molecular biologist as we go through the sort of system. But now where's the big excitement in terms of what we're doing? Well, I've already said at the moment we have a problem with bed nets. They're only beneficial if they're impregnated with insecticide, and pyrethroids are the only sort of insecticide that we've got. Now, when people started talking about putting pyrethroids on bed nets, I had a terrible tendency to go around asking people, what about resistance? How much resistance have you actually got out there? And again, if any of you know anything about insecticide, you will actually know that DDT and pyrethroids share a common target site. DDT is the insecticide that people have been using for 50-odd years, it's not inconceivable to think that there may be some target site resistance sort of sitting out there in the population after all those years of sort of hitting the population with the insecticide. If there isn't, in fact, the insecticide hasn't been working terribly well. That's the other side of the coin. But people would tell me, no, there's no problem out there. We don't have pyrethroid resistance. The reality was that people hadn't looked because, as I say, all the capacity for doing that sort of work had disappeared throughout Africa. When people did go in and look, it is quite clear that there is DDT/pyrethroid resistance that runs right the way from the top of the malaria belt in West Africa all the way down to Zambia. And that is one and the same resistance mutation that's in there, so that has spread over that sort of pocket. If I go over to the east coast, you actually find a much smaller pocket around Tanzania and Kenya of, again, DDT pyrethroid resistance, but it's a different mutation in this case, and in fact produces a lower level of pyrethroid resistance and a higher level of DDT resistance. And if you go into southern Africa, 5 years ago, they didn't have a problem. They switched 5 years ago to using pyrethroids for malaria control. Now, the reason they've switched back to DDT is that they do have a problem. And that problem is a very serious one. It's a very high level of pyrethroid resistance that's actually come through. We may be lucky, but we're only lucky temporarily in that the big tract of resistance that I've talked about down West Africa is probably not a high enough level of resistance to stop bed nets functioning. The resistance that is in southern Africa is high enough. Bed nets with pyrethroids are probably already dead in the water for funesters down in the southern end of Africa, and that's before they've been introduced. So we need to improve vector control as we're going through there. I haven't said very much about dengue yet. Well, the only thing we can do with dengue is actually go for vector control, because you've already heard from the virologist who started that dengue is one of those where we do not have a vaccine, and we're never likely to have a vaccine because we've got 4 serotypes, and as you've already heard, once you've had one serotype go through, when the next serotype comes through, if you get infected, the disease is worse. So the only current method of dengue control is going in and fogging with insecticide or removing the breeding sites, and that's tremendously difficult to do as you go through there. So what can we actually do to improve vector control? Well, I think the big hope that I and others have is that we can use the new genomics technology to actually allow us to develop new tools and technologies for this. Now, again, I've been working in this business for 20-odd years, and in those 20-odd years, we have had no new insecticides come into that sort of system for malaria or other vector control, and that's because the big ag companies do not develop things for vector control. They develop things for agricultural insects, and if they happen to work for mosquitoes, then great. If they don't, then tough. And it just happens that many of the newer insecticides that come through haven't worked terribly well for the vectors that we're actually coming through with. But we do only have a very limited number of insecticide resistance pathways that these things can go through. So what about add-ons that we could actually add to the insecticides to get them functioning? Well, here's just a picture of Anopheles gambiae polytene chromosomes. And here are where the 4 major resistance genes sit for DDT or pyrethroid resistance. We know what 3 of them are, and those are just the structural genes. Here's the sodium channel. These are glutathione transferases. These are P450s. This empty circle here that has no other arrows going into it is actually the major controlling resistance gene that if we could hit, we could knock out most of the resistance problem that we've got. That's because this controls the expression of these and of these as it goes through. I have no idea what this is. It's an empty circle. There are a couple of 100 kilobases of DNA sitting in there, and what gives you the extremes of that are microsatellite markers that we've been working with. But we now have a genome. That means within this region, I can pull out several hundred other microsatellite markers, and that means we can narrow down where that gene is from the genetic crosses that we've got, and I can then take candidate genes that sit in there, and using the transgenics, hopefully turn a susceptible insect into a resistant, and eureka, I know I've got my transacting factor that actually causes all these problems as I go through. I then have a target to go and hit, with something I can add into an insecticide formulation to actually work through. And I think that's where we can work with the genomics and put something novel into the system. What else can we do? Well, we've already got the technology coming through for insect transgenics. We can actually already devise molecules that will block dengue and malaria. We can put them into insects. We can put them upstream of promoters that are set off with the blood meal, or when the insect takes a blood meal in. So when your insect takes its blood meal with a parasite or the virus in, those genes start expressing like crazy, actually in the gut. And the first thing that that parasite or virus hits then are cells which actually should be able to kill it as it goes through. Now, that was all sort of pie in the sky 10 years ago. We didn't have the transgenic insects, and we didn't have the ability to do any of that. We do now. It's coming. Some of that is already published. Where are the big sort of stopping points now within the system? Well, I've already given you a demonstration of how difficult it is just to get a basic diagnosis of malaria or anything else done out in the field at the moment. Are transgenic insects going to be any better? Well, I guess the one positive that they've got, if you can get them out there and you can replace the population, is that they really don't care where they're flying, whether it happens to be to a poor man's house or a rich man's house. They will spread and do the job for you, again, if you get the technology right. So we've got a technology that's independent of the poverty or many of the other economic factors that we've got around. But, and there always has to be a but in these sort of stories, we don't yet know enough about the population biology of these insects to know how quickly we can spread them. In fact, the dengue may be the first one we can have a shot at, because I've said there we've got one vector. Now, for each of these transgenic systems, you're going to put that gene into one kind of insect. Well, we can get those sort of genes into Aedes aegypti now. We can start and look at cage population replacements and see how those work and how these genes spread through populations. We now have to do the social science. We now have to do the politics. We have to make sure that the scientists and the people in developing countries are happy with the technology, understand the technology, can work with the technology, and want to move ahead with us actually using this. And these are not simple technologies for people to understand or work with. And there's another divide there in terms of saying, how are we going to get people involved in this? How are we going to get people understanding what you can do with this and how you can do it? So I don't think we underestimate where we're going with some of these technologies, but at least they're there as potential new technologies as we go through. If dengue comes through as the first set, then obviously we've got malaria and the anophelines following that. We have the technology now coming through to be able to put these genes into Anopheles mosquitoes. We've got the first transgenics working in Anopheles gambiae and Anopheles stevensii. It's not going to be easy in all mosquito vectors. This is difficult technology. Again, to put this into context, I said you've got two major vectors in Africa. Others may disagree. They may say you've got more than that, and in fact there are certainly more vectors than that around. The first colonization of Anopheles funestus occurred last year. We do not have transgenic technology for Anopheles funestus. We will not have transgenic technology for Anopheles funestus for quite a few years to come, because we understand far less of the biology of that beast than we do of gambiae and of others. So again, I haven't got a magic bullet. Nobody has. But we do have some technology coming through. We've talked about the divide between the social scientists and the molecular biologists as we go through. And we've also talked about the Luddite fringe and those of us who are the geeky techs on the other side, and how we actually sort of put those two together. But I think there's also another dangerous divide that I want to flag up, and another set of pressures that we need to be very careful of, because as these technologies come in, and people like me and others get very excited about what we might be able to do, and we get some very good postdocs and things coming in and working with those, we compete for funding that comes through the Medical Research Council or the Wellcome Trust or NIH or others, And we're competing against people who are working on cancer or other sort of sets like that. So the science we've got has to stand up to those levels and compete at that level. The postdocs that we've got coming through those labs have to get their career tracks going, which means they've got to publish in the likes of PNAS and Nature and others, because if they don't, they're not going to get their tenured positions as they go through. That's great if all I had to do was do that work in a developed country lab and we could carry on in our ivory tower and get that running through. But I actually need to get this into the field in 5 years or 10 years' time, and that means developing country scientists coming along track there. That also means they're generally starting at a much lower base with the molecular side. They have a competing set of requirements that needs them to come up to a certain level. And I have to get the postdocs in the lab to help them do that, which could be a downside for the actual sort of career tracks of those individuals who are in there. How do you balance that? How do you balance the fact that the science may not move as fast as you might do within cancer or other sets there? So I think there's another competition going on within that set that we all need to be very careful of. And it's great to see malaria back on the agenda with a lot of the other infectious diseases. But before we all get carried away with the hype, let's make sure that we get that message across to the politicians and the grant funders, because in 5 years or 10 years' time, if we haven't got done quite what we might have done because we've been trying to cope with a lot of these competing pressures, we need to make sure that we've said that up front, that the timescale was going to be longer because we had to do various things, and that we don't run alongside some of the others. That, I guess, is just a sort of finishing message as we go through. But I think there are lots that can be done with the technology. I've concentrated on the vector side and given you some examples there. But it's clear on the parasite side that we've got new targets, new drugs, new things that could come through here. We've still got the same problems in delivering. So at the end of the day, we're not going to solve all the problems overnight in any of these sort of sets. We're all in here for the long haul. The same is true of vaccines. And again, many of you, I'm sure, will have been to waiting for the vaccine meetings. I think the first waiting for the vaccine meeting I went to was in 1978, when they told me in 5 years' time we'd have a vaccine. And I think every 5 years since, I've been to a waiting for the vaccines meeting where the vaccine's going to be ready in 5 years' time. Now, I think there are lots of prospects there, but it's not going to be short-term as we go through. And so I guess that's the parting message that I have for the audience. Thank you.
So there are many challenging questions that were raised. I open the presentation for discussion. Yeah.
Thank you. Andrew Moore, EMBO. I have a question about the biology of the malarial parasite in the mosquito host. Is it a commensal when it's living there, or is it a parasite? And would the mosquito benefit if the parasite were killed when it ingests it? In other words, could introducing resistance to it in the population lead to a sort of positive feedback where it would then be positively selected for and possibly spread more widely? Okay,
simple answer to that, I think, for malaria is that we've got no real evidence that there's a huge negative impact on the mosquito from having the malaria parasite in there. That's not true of something like filariasis, where the microfilaria, once it gets in and develops to an L3, causes enormous damage to the mosquito when it gets upstream and has to sort of burrow its way out of the mosquito's head. But the malaria parasite itself seems to cause very little damage to the mosquito, and so the mosquito just tolerates it being there. There are 1 or 2 mosquitoes out there that are naturally resistant, and a lot of the kind of resistance genes that we're talking about putting into mosquitoes are actually coming from mosquitoes that are naturally resistant. So there's a lot of work going on to identify those natural resistance genes. But the parasite's clearly evolved to a point where it's not damaging its host as far as the mosquito is concerned. So we're not going to get that as a driver to push things through the population. I think if that were the case, it would probably have happened naturally by now.
Thinking along similar lines, Mario Colucci used to speak about the possibility of persuading female mosquitoes to become autophagous like their male partners and sort of breed some transgenic dominant negative that just wouldn't feed on people. and will come to sexual maturity without having a blood meal. Is this pie in the sky, or is it worth pursuing?
I think it's worth pursuing. I don't know anybody who's actively pursuing it at the moment. I mean, we do have some strains of mosquito that are autogenous that will go through the first egg-laying round without taking a blood meal. But that's a Culex. I don't know of any Anopheles mosquito that will do that. So it's clearly possible to do. I don't know anybody who's actively chasing the genes to actually that actually determine what it is within the Culex that do that. But I may be wrong if anybody else knows of anybody doing that.
I was very interested to hear about the cord blood transfusion, and I'm going to fantasize. The fantasy is that if you do this, you'll create chimeric individuals because of the enrichment of the stem cells in cord blood. And it is possible, according to some extreme immunologists, that that's a very good way to set up individuals for autoimmune disease, which may be a relatively small price to pay. But do you have any thoughts on that?
I was going to say, it's kind of one of these sort of setups, isn't it? You know, it may cause problems like that, but if you're going to be dead anyway in 24 hours, are you going to worry hugely about it? And I guess the answer is probably not. The other side of doing that, I guess, would, if you could set the system up in such a way, you would save the cord blood from that individual as it was born. And if the individual then went down with malaria or something else and came back into the hospital, the first set of blood that you would use to treat it would be its own.
This is very common now in America,
yeah.
where people freeze cord blood as a gift to their children. But I think it might be very worthwhile to at least consider the idea of going back and seeing whether you have created chimeric individuals. It would be useful to know if that's going to be a problem.
Yes?
Coming back to the society aspect, Bertolt Rutz from European Patent Office. Coming back to the society aspects, with the background of African governments refusing gene-modified food for starving population, do you see any chance that ever it would be possible to release transgenically modified mosquitoes in Africa, not to speak of all the NGOs in the developing world who think they can decide for in the developed world who think they are up to decide for the developing world what could be good for them. Could you comment on that?
Okay. I think it's curious that personally I haven't hit as much resistance to this idea as to the food sort of setup, which is curious. I mean, I sat on a kind of any questions type panel, and there was an awful lot of resistance against the genetically modified foods, people saying we don't need sort of riper tomatoes or whatever, but far more of an understanding, I guess, that you were doing something positive about a disease that was extremely intractable as it goes through. That isn't to say I underestimate the problems. I mean, we all, or maybe we don't all know, but using genetics to try and control disease is not new. In fact, there was a whole big program of sterilizing mosquitoes and using that as a genetic control sort of system back in the 1960s and 1970s. And that went on in India and various other places. And there was a big scare story that got around in India that said they're not actually sterilizing these mosquitoes and doing them as sort of sensible genetic control. They're trying to put something nasty into the population with these insects, and they kick the whole program out. So you clearly do have to be careful of the politics as they go through, and we've got experience of that. And that, I think, is exactly why there is no point me or anybody else working in an ivory tower on our own. We have to do this in combination with the developing country scientists. We have to do this with the governments of those countries as we're going through. And at each step in the development, everybody needs to be involved, because there's no point me or anybody else going in 5 or 10 years' time to South Africa or Mozambique or Zimbabwe or Tanzania or Benin and saying, we've got the great magic bullet for you. And in fact, if you do go for one of those sort of big vectors, which we will eventually, they don't respect country borders either. So I guess it's an even bigger problem than that you can't just release in Benin and expect, you know, that you're going to cure the malaria in Benin, but those transgenic mosquitoes are going to stop at the border and present their passports. You know, it isn't going to happen in that sort of sense. So I think it's not one government that we've got to get on board. We're looking at a very large-scale sort of program here. Is it technically feasible? I think the positive end of it is that, again, the sort of technologies that we're looking at, although we'd be putting a foreign gene, even if we may be taking it out of a mosquito and putting it into another mosquito, into those populations, are that we know that the sort of drive systems that we're talking about working with do sweep through insect populations. We've got evidence from Drosophila in looking at P-element sweeps that go through populations of Drosophila worldwide. So there is the potential for it working. I think the political obstacle may be harder than the science at the end of the day if we're not careful. And I think that's why we need to start working on it now, not when we've got every T crossed and every I dotted as far as the science is concerned.
Yeah, I just have one silly question. The first one, what does on your slide of the genome, what does QTL stand for?
Okay, quantitative trait loci.
Right.
I'm sure you're now sort of entirely
really
clear as to what that is. I didn't know what a QTL was either.
I'm Tom Clark from Nature magazine.
For many years. I mean, basically what we're looking at, or we're saying, is that there is a trait within that bit of DNA that is encoded within that sort of set, and we don't know what it is. So we're not calling a gene because it could be all sorts of things. It's a sort of very broad term, basically.
And secondly, you mentioned insecticides and the fact that, and this I know to be true, that it's big ag companies obviously have no interest in controlling disease vectors. But surely insecticide development compared to, for example, developing a vaccine against malaria is quite cheap, I imagine. Do you know of any efforts to fund investment in new chemical technologies to control mosquitoes? I've not heard of any. Do they exist?
I think you'd be wrong in saying developing an insecticide is cheap. I think the amount of money that the ag companies would now quote at you because of all the regulation that they have to go through in terms of human safety and tox data that they have to put together as a package is from start to finish, i.e. getting your product as a molecule that works to actually getting it out onto the market, takes about 7 years and is about $20 billion. So it's not cheap, and it's not what you would give a small company to do. There is a public-private partnership which is actually at the moment funded by NIH on a small set. I believe there are 5 of us who have grants, and I say us because I've got one of them, working with some biotech companies. What we're not doing is trying to sort of develop new insecticides from scratch. What we are trying to do is take what's been developed for the agricultural side and do enough modification that we can get it to work for the vector populations. And that actually gets rid of some of the problems on the tox package that needs to be put through, although not all of them, as you go through. And the same is true if you go for an add-on to an insecticide that's already there. But I think everybody will tell you, starting from scratch to design an insecticide for a vector is a non-starter. It's economically just not viable.
Okay, I think we'll stop here. But you know, there were 2 questions, and I give you the priority.
Anyway, what I wanted to say that the problem with African leaders accepting the genetically modified mosquitoes doesn't exist because currently the International Atomic Energy Agency is working with African governments using the sterile insect technique to control or eradicate the tsetse fly as part of an integrated area-wide pest management program. But the problem here is not so much the African government's willingness, acceptance, because they have actually endorsed this eradication campaign. The problem is with the donors, because the donors are arguing that if you eradicate, what are the ecological effects? So it's not the African governments. I don't think they have a problem.
Okay, I think you've got a very different setup there. You're talking about a sterile insect, so that doesn't sort of reproduce itself. You go in and you wipe the sort of system out that way by sterilizing a very large number and swamping the population that's already there. I think a genetically modified organism is something different because it does reproduce itself and it's designed to do that. And it's going to stay out there in the population, and it's going to mutate and sort of generally sort of replace the population permanently. So I think you have very different systems, but I would agree the very fact that you can go in for a mass sterile release, or in the past we've been able to go in for genetic control sort of systems and got agreement from various governments, bodes reasonably well for being able to put this technology out there, as long as we work very sensibly with the governments to do it.